Many people think that war and slavery are modern political failures unique to humans, but science shows they are actually ancient survival habits inherited from our animal ancestors. For billions of years, living creatures had to fight for scarce food and land to survive, which created a sort of built-in survival software in nature. For example, chimpanzee groups will violently attack their neighbors for territory, and certain ant species actually raid other nests to steal babies for slave labor. Today, we often blame modern conflicts on secret plots or bad leaders, but the real cause is much deeper. War and cruelty still happen because human society allows unchecked power structures to grow, and because communities pass down survival trauma for generation to generation. This paper presents an evolutionary, game-theoretic, and sociological framework, positing that contemporary socio-ecological collapse is not an accidental byproduct of human nature, but a structurally and psychologically manufactured crisis. While standard discourses conceptualize warfare and systemic exploitation as localized political failures, an examination of evolutionary biology reveals them as deep-seated survival mechanisms or legacy software shared with taxa such as pan-troglodytes, coalitionary territorialism, and Formicidae, doulosis. For approximately 95% of human history, Homo sapiens operated within sustainable ecological feedback loops that suppress the worst of these drivers, reinforcing our baseline nature as a biological eusocial species embedded within a self-regulating planetary superorganism, the Gaia hypothesis. The modern crisis arises from unregulated hierarchical structures fueled by transgenerational survival trauma. Insulated by hyperwealth, elites employ hyper-optimized survival strategies that suffer from a fatal evolutionary blind spot. Their prehistoric mammalian programming drives endless power accumulation, blinding them to the mathematical reality that they are destroying their ecological host. To sustain this trajectory, elite power structures weaponize state-level psychological warfare, exploiting the masses by depleting their cognitive energy on fabricated culture wars over arbitrary identities and institutionalizing dogmatic religions to codify rigid hierarchies. This represents a profound rupture from ancestral animistic belief systems, which historically functioned as stabilizing ecological feedback loops. Consequently, global systems are maintained by two distinct, unconscious groups: distracted masses and blind elites, both prisoners of prehistoric biology, operating in an industrialized world. This study demonstrates that replacing political figures is futile, as any new leadership cohort inevitably succumbs to the same automated mammalian dominance program. The ultimate systemic solution requires a breaking in this evolutionary loop through the global prioritization of mental health. By systematically treating the human mind as critical planetary infrastructure, resolving collective transgenerational trauma, and regulating survival-driven nervous systems, humanity can transcend automated biological competition and realign with the self-regulating egalitarian harmony of the planetary superorganism. Anthropological and sociological discourses frequently conceptualize warfare and systemic human exploitation as culturally localized pathologies or modern sociological political failures. However, an examination of evolutionary biology reveals that intergroup aggression and resource-driven exploitation are not unique to Homo sapiens, nor are they modern anomalies. Biological analogs such as lethal coalitionary territorialism in pan-troglodytes and systemic pupae raiding, doulosis, among Formicidae demonstrate that these behaviors represent ancient behavioral adaptations optimized for resource security under ecological scarcity. Originating deep within life's 4.2 billion year evolutionary trajectory, these survival mechanisms constitute a biological legacy software. In modern human populations, the persistence of these destructive dynamics is often misattributed to localized conspiracies or fixed human architecture. In reality, they represent the systemic manifestation of unregulated hierarchical structures driven by deep-seated transgenerational survival trauma. The deep time paradigm versus the anthropocentric anomaly. In the study of evolutionary biology, conflict is recognized as an immutable foundational catalyst for structural complexity, originating roughly 3.5 to 3.8 billion years ago, alongside cellular life itself. For approximately 85% of Earth's biospheric history, this evolutionary warfare operated on a strictly microscopic scale, devoid of conscious intent, teleology, or strategic foresight, progressing instead through immense chronological timelines via random genetic mutation and passive environmental selection. This deep time dynamic stands in stark, catastrophic contrast to human biological warfare, a dangerous anomaly emerging only within the last 4,000 years of the late Holocene. While natural macroevolution relies on co-evolutionary balance and long-term genetic counteradaptation, human weaponization condenses genetic manipulation and pathogen deployment into single generations. By bypassing natural selection to engineer rapid, intentional destruction, anthropocentric biological warfare violates the systemic checks and balances built over billions of years of host-pathogen co-evolution. Understanding the sheer depth of this evolutionary timescale is the critical first step in diagnosing modern humanity's divergence from systemic planetary stability. The chronological timeline of deep time warfare. This ancient biological warfare unfolded across major geological eons, beginning in the Archean eon between 3.8 and 2.5 billion years ago, when Earth's early oceans were dominated by single-celled anaerobic prokaryotes operating under intense resource scarcity. Initial evolutionary conflict manifested passively as chemical scavenging and raw metabolic competition for limited organic compounds. But the subsequent emergence of virulent bacteriophages introduced the planet's first true predatory genetic entity. These early prokaryotes faced recurrent extinction events from viral lysis, forcing the development of the earliest genomic countermeasures to prevent total population collapse. As life transitioned into the Proterozoic eon between 2.5 and 0.54 billion years ago, the metabolic proliferation of cyanobacteria resulted in the catastrophic accumulation of atmospheric oxygen. This great oxidation event triggered a planetary mass extinction that forced radical cellular adaptations and the evolution of antioxidant defenses. Concurrently, predatory conflicts shifted through phagocytosis, ultimately evolving into mutualistic endosymbiosis and giving rise to mitochondria, while pathogens evolved sophisticated mechanisms to subvert host clearance by surviving inside host cells rather than causing immediate lytic destruction. It was during this eon that inside host cells and meiotic sexual reproduction evolved, primarily as a mechanism to continuously reshuffle genetic alleles so that rapidly evolving parasites could not lock onto static host genotypes. Finally, the Phanerozoic eon, spanning from 541 million years ago to the present, initiated the Cambrian explosion, which rapidly diversified multicellular body plans and established high-density host populations. This shift allowed pathogens to transition from targeting individual cells to exploiting complex, specialized organ systems, such as gastrointestinal, respiratory, and vascular networks, while the emergence of mobile macrofauna created dynamic vectors that enabled pathogens to travel vast geographic distances and execute cross-specific zoonotic jumps. Generational velocity and asymmetric kinetics. The core engine driving this deep-time molecular conflict is a profound kinetic imbalance in reproduction rates between host organisms and microscopic pathogens. On one side of this kinetic divide lies microbial hypervelocity, categorized by prokaryotes like Escherichia coli, undergoing logarithmic binary fission every 20 minutes under optimal conditions. When combined with viral burst sizes that yield tens of thousands of progeny within hours, and the error-prone polymerases that guarantee millions of unique genetic variants per day within a single host, pathogens possess an astronomical evolutionary speed. Conversely, complex multicellular macroorganisms are constrained by host inertia, requiring months, years, or decades to reach reproductive maturity. While facing a severe temporal lag because adaptive mutations can only be passed to subsequent generations via a strict germline bottleneck of sperm and egg cells. To survive this extreme asymmetrical kinetics, complex hosts evolved a localized somatic solution that mimics microbial speed within an individual's timeline. Through VDJ recombination, the vertebrate adaptation immune system utilizes targeted genetic cutting and error-prone repair to generate millions of unique antibody variations, successfully fighting microscopic velocity with internal cellular velocity. Molecular mechanisms of ancient warfare. This historical record of this multi-billion-year kinetic battle is permanently etched into the architectural landscape of modern genomes. Within prokaryotic defense suites, restriction modification systems act as primitive molecular scissors, utilizing restriction endonucleases to recognize and cleave foreign viral DNA as specific sequences while protecting host DNA via defensive methylation. Furthermore, bacteria maintain an adaptive immune memory through CRISPR-Cas systems, which function as a literal molecular archive of historical conflicts by capturing fragments of invading viral DNA, integrating them into the host genome as spacers, and deploying them as RNA-guided sequence-specific mechanisms to neutralize subsequent viral invasions. When these defenses fail, prokaryotes utilize abortive infection systems, a form of evolutionary altruism where a cell undergoes programmed suicide upon detecting an unmitigated viral infection, sacrificing itself to protect the surrounding clonal colony from viral propagation. Eukaryotic genetic archaeology reveals similar scars, most notably in the form of endogenous retroviruses, which are ancient viral epidemics that successfully infiltrated the germlines of ancestral species and became permanently trapped within the host genome, now comprising approximately 8% of human DNA. Over evolutionary time, hosts have progressively exapted these genomic fossils for vital physiological function, exemplified by the protein syncytin-1, which is absolutely critical for human placental morphogenesis, but was originally an infectious viral envelope protein captured from an ancient retrovirus. The laws of natural conflict, the Red Queen, and equilibrium. Ultimately, the dynamics of natural biological conflict are governed by strict systemic laws, primarily Van Valen's Red Queen hypothesis, named after the character in Lewis Carroll's Through the Looking Glass, who remarked that it takes all the running you can do to keep in the same place. This principle dictates that neither host nor pathogen can achieve a permanent evolutionary victory, as any defensive advance in host immunity triggers an immediate survival-driven counter-adaptation within the pathogen population, locking both into a continuous loop of co-evolutionary pressure. Crucially, this natural evolution possesses no teleology, conscious strategy, long-term blueprint, or moral framework. Genetic variations occur purely through stochastic copy errors, radiation, or mobile genetic elements, while natural selection operates merely as a passive retrospective filter that conserves traits facilitating immediate survival while ruthlessly eliminating non-viable biological strategies. This non-teleological process naturally guides stable host-pathogen relationships toward a dynamic equilibrium rather than total destruction. Under the avirulence hypothesis, pathogens that cause 100% host mortality exhibit low evolutionary fitness because they rapidly eliminate their own transmission vectors and burn out, meaning that deep time naturally favors moderated evolutionary equilibrium. And it also favors moderated, balanced virulence optimized for transmission. Unmitigated mass lethality is therefore rarely an evolutionary equilibrium, but rather a symptom of novel, unadapted cross-species jump, a biological reality that exposes just how severely human biological warfare diverges from the natural order. The architecture of reciprocal genetic adaptation. The co-evolutionary arms race serves as the primary operational engine of biological conflict, transforming the evolutionary landscape from a static physical environment into a living, fluctuating matrix, where the host and pathogen continuously redefine one another's selective pressures. Unlike adaptation to fixed geological or climatic conditions, any defensive innovation arising within a host population exerts an immediate, existential selective pressure upon the competing pathogen, mandating a reciprocal counteroffensive to avoid total extinction. This dynamic establishes an unyielding, hostile feedback loop that progresses through a distinct, mathematically trackable kinetic sequence. When a random genetic mutation alters a host surface protein and effectively blocks pathogen attachment, This advantageous allele triggers a rapid selective sweep, proliferating across successive generations as non-resistant individuals are systematically culled from the breeding pool. This shift precipitates a catastrophic collapse in the pathogen population, as its primary mechanism for cellular infiltration and metabolic replication is neutralized. However, this extreme demographic bottleneck imposes an intense selective pressure on the surviving viral or bacterial reservoir. Any rare mutant variant possessing a modified building ligand that matches the host's altered surface architecture will experience an immediate fitness advantage. This adapted pathogen strain then undergoes a rapid resurgence, swiftly decimating the previously secure host population until a novel host mutation arises to reset the cycle. Molecular mechanistic battlegrounds. This reciprocal genetic warfare manifests across highly specialized biochemical arenas, predominantly focusing on the kinetics of cellular entry, intracellular stealth, and metabolic exploitation. At the cellular perimeter, conflict is dictated by a structural lock and key dynamic governing receptor-ligand interactions. A premier example of this is the evolutionary manipulation of transferrin receptors, TFR1, in vertebrate hosts. While mammals utilize transferrin to safely sequester and transport iron through the vascular system to starve out invading microbes, bacteria have developed specialized surface proteins designed to hijack TFR1 and siphon this vital nutrient. Over generations, mammals continuously alter the outer surface configuration of TFR1 to disrupt bacterial binding without compromising oxygen transport, forcing bacteria to continuously reshape their matching ligands to retain access to the host's iron reserves. A similar molecular dance occurs during sialic acid manipulation, where viruses such as influenza deploy the surface glycoprotein hemagglutinin to bind to sialic acid sugars on host cell membranes. In response, hosts have evolved complex variations in their cell surface sialic acid structures alongside the secretion of decoy sialic acids within their mucosal barriers, effectively trapping viral particles in a molecular matrix before they can reach and penetrate the underlying cell layer. Once a pathogen successfully breaches these outer defenses, the battleground shifts to an intracellular conflict between stealth and detection, pitting host pattern recognition receptors, PRRs, against microbial pathogen-associated molecular patterns, PAMPs. Host cells utilize specialized PRRs, such as transmembrane toll-like receptors, to scan the intracellular environment for conserved, immutable microbial signatures, including viral double-stranded RNA or bacterial flagellin. To counter this internal surveillance network, pathogens have evolved a diverse arsenal of molecular antagonists, proteins synthesized exclusively to degrade, competitively block, or structurally mimic host PRRs, effectively blinding the host cell's internal alarm systems and delaying an interferon response. Concurrently, this conflict extends into chemical warfare via metabolic exploitation and the potent toxins of antibiotics to actively destroy host tissues or eliminate surrounding microbial competitors. In response, targeted organisms have developed sophisticated ATP binding cassette efflux pumps, which act as molecular bilge systems that actively reject these toxic chemical compounds from the cytoplasm back into the extracellular space before races are vividly captured through long-term ecological field studies where genetic variations in both host and pathogen populations have been longitudinally tracked. A classic example is the intentional introduction of the myxoma virus into Australia in 1950, a biological intervention designed to eradicate an invasive and ecologically destructive population of European rabbits, Oristolagus caniculus. The initial viral strain was exceptionally virulent, inducing a 99.8% mortality rate among infected hosts within days of exposure. However, this extreme lethality imposed an unintended evolutionary bottleneck on the virus itself, because infected rabbits died so rapidly. Vector insects, such as mosquitoes and fleas, lacked a sufficient temporal window to bite live hosts and transmit the virus to new populations, driving highly lethal viral strains toward an evolutionary dead end. Concurrently, the surviving 0.2% of the rabbit population bred rapidly, passing down vital genetic resistant variants concentrated in interferon pathways, such as the OAS1 gene and the PRNP locus. Over subsequent decades, natural selection penalized unchecked lethality and favored attenuated, moderately virulent viral mutations that kept the hosts alive longer, thereby maximizing the transmission window. This dual adaptation steered the system away from mutual destruction and toward a stable dynamic equilibrium, characterized by highly resistant rabbit populations coexisting with an optimized, less lethal viral strain. In botanical systems, this co-evolutionary balancing act is perfectly illustrated by the wild flax Linum marginale and flax rust Melamprosa linni system, which provided the empirical foundation for Harold Henry Flor's seminal gene for gene hypothesis. Within this ecological framework, host resistance genes directly corresponded for pathogen avirulence genes in a highly specific spatial mosaic. When a wild flax plant possesses a specific gene, its intracellular receptors successfully detect the matching protein secreted by the invading fungus, immediately triggering a localized hypersensitive response, a form of targeted cellular suicide that desiccates the local tissue and starves out the biotrophic fungus. If the fungus undergoes a mutation in its gene that alters the shape of its secreted protein, it effectively evades host detection, allowing it to successfully parasitize the plant. Because both host and pathogen populations exist in geographically fragmented pockets, this constant reciprocal genetic matching prevents either the wild flax or the rust fungus from ever achieving absolute dominance, cementing a state of fluctuating polymorphism that maintains a long-term biodiversity through perpetual, localized conflict. The genetic and energetic fitness costs. Crucially, neither the host nor the pathogen can deploy an infinite or flawless array of defensive or offensive biological mechanisms, as every evolutionary adaptation is strictly governed by metabolic allocation constraints and energetic trade-offs. Organisms operate under a finite energy budget. Any resources diverted to manufacture complex cellular defense suites, synthesize secondary metabolites, or maintain hypervigilant immune responses are fundamentally stolen from the metabolic pathways dedicated to somatic growth, tissue repair, and reproductive output. This allocation problem introduces a severe fitness drag on both sides of the evolutionary conflict when the immediate threat is absent. This inescapable biological tax ensures that natural evolution never produces a permanent victor. Instead, it maintains a systemic check and balance architecture, preventing any single species from achieving the unmitigated runaway exploitation that characterizes human industrial and biological deviations from Gaia's dynamic equilibrium. The mathematics of equilibrium and the virulence trade-off. In natural ecosystems, biological conflict rarely culminates in the absolute extinction of either host or pathogen. Instead, deep evolutionary time systematically guides both actors toward a state of dynamic equilibrium, governed by the cold mathematical reality of the transmission-virulence trade-off model. For a parasite or pathogen to successfully propagate its genetic lineage, it must optimize its survival within the biological vehicle responsible for its transmission, the host. The ultimate metric of evolutionary fitness in epidemiological systems is the basic reproduction number, which defines the average number of secondary infections generated by a single infectious host within a completely susceptible population. This relationship is quantified by the classical deterministic epidemiological equation. Within this mathematical framework represents the transmission rate or probability of host-to-host transfer given contact, denotes the absolute number of susceptible individuals in the host population, represents the host recovery or immune clearance rate, is the virulence defined specifically as the pathogen-induced host mortality rate, and is the baseline natural mortality rate of the host population. The functional constraints embedded in this equation introduce a severe fitness tax on unchecked pathogenicity. As virulence scales linear, the denominator increases, which sharply reduces the duration of the host's infectious window. To maximize the transmission rate, a pathogen frequently requires elevated within-host replication rates to increase shedding volume via respiratory droplets. Shedding, or viremia, however, this aggressive viral or bacterial load inherently accelerates host tissue damage, driving up. Natural selection continuously filters out mutant lineages that maximize individual cell lethality at the expense of population-level dissemination. Instead, it optimizes for an intermediate fitness peak, the exact infection point where the maximum transmission velocity is achieved immediately before the host is immobilized or killed, thus preventing the breakdown of the infectious chain. Transmission pathways as drivers of equilibrium kinetics. The exact evolutionary baseline of this equilibrium is determined by the pathogen's specific mode of transmission, which dictates how much somatic damage the microbe can afford to inflict on its host without disrupting its own reproductive lineage. Pathogens requiring direct contact or active host mobility to spread, such as rhinoviruses, influenza variants, and other sexually transmitted infections, face intense selective pressure to downregulate viruline. If a directly transmitted respiratory virus induces rapid prostration, immobilization, or mortality within hours of infection, the host is confined to bed and physically isolated from public spaces. This behavioral restriction instantly disrupts the transmission chain, breaking the path to a sustainable unit. Consequently, natural selection favors mild, chronic, or asymptomatic clinical phenotypes that preserve host mobility and maximize long-term environmental shedding. Conversely, vector-borne pathogens that utilize external arthropod agents such as Plasmodium, falciparum carried by mosquitoes, or Yersinia pestis carried by fleas, are largely uncoupled from the physical mobility constraints of the vertebrate host. These organisms can afford to render the vertebrate host severely comatose or bedridden. A completely immobilized, lethargic host is a highly vulnerable, defenseless target for biting vectors, directly elevating the significantly higher natural equilibrium virulence thresholds than direct contact pathogens. Finally, pathogens characterized by prolonged environmental persistence, such as Bacillus anthracis spores or Clostridium botulinum, bypass the requirement of an intact living host chain entirely. These organisms can survive in soil, water, or detritus for decades, enabling them to kill the host rapidly, return to the substrate via carcass decomposition, and remain metabolically dormant until encountering a subsequent susceptible host. In these instances, natural selection treats the host not as a partner for long-term symbiosis, but as an ephemeral, highly destructive resource to be rapidly spent. The avirulence hypothesis and its nuances. The historic avirulence hypothesis, engineered by early comparative pathologist Theobald Smith, posited that all symbiotic and parasitic relationships inevitably evolve toward benign coexistence because harming the host is inherently counterproductive to the pathogen's long-term survival. While modern evolutionary biology has replaced Smith's linear teleology with the more nuanced trade-off model, the fundamental mechanics of his observations regarding novel encounters remain highly accurate. Extreme explosive mass lethality is rarely an optimized evolutionary strategy. Rather, it is almost exclusively a symptom of unadapted evolutionary recent cost species jump, or zoonosis. When a pathogen spills over from its natural reservoir host, with which it has achieved a deep time equilibrium over millions of years, into an accidental immunologically native host, the regulatory biochemical signaling cascades completely break down. The pathogen replicates uncontrollably or triggers a hyperinflammatory immune overreaction known as cytokine storm, killing both organisms in an evolutionary mismatch. Over vast timelines, persistent selection pressure systematically clips these lethal, unadapted extremes from both host and pathogen lineages. On the host side, individuals possessing hyperreactive or completely defenseless genetic profiles are weeded out of the gene pool. Concurrently, the pathogen population favors mutated variants that downregulate intra-host replication or employ molecular camouflage to evade immune detection without causing systemic shock. This long-term pruning of biological extremes transforms chaotic host-pathogen conflict into stable commensalism and can ultimately lead to obligate mutualism or genetic endogenization. This is the exact process by which ancient aggressive genetic entities were structurally integrated into host biology, mirroring how my unified hypothesis views human eusociality as our true, uncorrupted architecture currently masked by transgenerational survival markers.